thanks everyone for joining. We don't want to take up too much of your time, so I'm just going to share my slide again. This is about our spatial transcriptomics data analysis in Python workshop. And we kind of put spatial transcriptomics data in there. But I think nowadays we more or less even like to call it spatial omics, because of course a lot of modalities are not only transcriptome based, and a bunch of these things actually also going, of course, very much the protein targeted side and so on. So I think these tools getting more agnostic, but also always sort of focusing on this multimodal aspect of things. Let me just, so my name is Fabian Theis. I'm from Helmholtz Munich and Technology of Munich. I've been working in sort of developing machine learning tools in single cell field in particular for quite some time. And we're jointly organizing this workshop together with the Single Cell Omics Network Germany that I co-coordinate with a bunch of friends from all over Germany. And we're really happy that we have a bunch of fantastic presenters and tool developers doing this workshop over the next 2 days, and in particular sort of having this really strong focus on PhD students developing these tools and then actually pushing them forward. I'm super excited about learning about that. Just super briefly, oh yeah, I made a fantastic animation. I hope you can see that, just to sort of get your attention a little bit and to motivate maybe a little bit what like a live thing would be, that I think could be more fun. But I guess that's maybe at least the simplest way to do such an 8-hour workshop from people joining all across the world. Briefly, just one slide about this single cell omics idea. There we wanted to really bring together single cell researchers and strengthen the community. Initially, we've been obviously focusing sort of more on the national thing, but with all these virtual events, I think this has been becoming a bit bigger, and it's actually really exciting getting so many people interested in that. I think we had really for some events more than a few hundred people joining, and it's really exciting. We want to promote development, use of single cell technologies, exchange, computation, experimental methods, expertise. This is essentially today about computational methods in single cell analysis. Of course, also connect to the relevant international initiatives such as ACA and so on. This network. These are partners, in particular, Nikolaus Rajewski and Jörn Walter's lab, been pushing this with us now with Joachim Schulze, many others, Olli Stiegler now particularly being very active, Maria Elena and Barbara Treutlein being sort of steering and helping coordinate this, maybe do proper thing. We had already a related workshop a year ago. Well, some of you remember that. This was actually a talk-based workshop with people presenting about their most recent techniques of spatial analysis, but also for spatial assays, as well as for the computation analysis. It was a really fun event, I would say. And I think what's exciting today now is that we want to now speak more about, once we have a lot of data, how do we maybe sort of bring analysis into a workflow, sort of a workflow that we're more used to maybe from the single cell disassociated side, where we really have very clear steps and put things together. And I think things have been quite advancing over this past year, so I think it's a good time. So what we thought, what could be interesting tools, and I'm going to comment a little bit about why we chose these in a minute, were those four. We want to talk about squidpy, set location, eggplant, and ncem. And yeah, we're going to do this in 2 sessions, 2-3 hours, 2.5. These were actually 2-3 hours, because we actually also have talks today and tomorrow, and we have presenters. And this is, I think, really cool. Basically, PhD students: Giovanni, Alma. We have Vitalii, Giovanni, Anna, and David from Helmholtz. Alma from Sweden, Lundberg lab, and Vitalii from Olli Stiegler and Uwe Barakta's lab. And they're essentially talking about their tools, and then sort of hopefully trying with a bunch of hands-on notebooks getting you into these topics. Schedule is, after my brief introduction, talking about squidpy, then about the cell-cell communication analysis with this ncem today, just having open Q&A. And we've been really initially, right, Giovanni, Anna, we've been initially planning to have sort of breakout session, people asking their very specific questions on the one data set, but at some point realizing that this would not scale to that dimension. So I think instead of these breakout sessions, we now have a asynchronous chat and trying to get support from the lab, from various labs, and actually to get that, actually working on your data. Is that roughly correct?
Correct, yes.
Please, everyone, give us feedback if you think this works or not. I think this is really important. We really want to see what you guys think, if that's okay or not. Second day will be followed by Alma's presentation on eggplant. And Alma, I don't know, I haven't heard you talk about that. How did you come up with that name?
It's an acronym. It's for effortless generic GP landmark transfer, but I also sort of like to eat eggplants. So that's actually the name, its origin.
It eats eggplant? What?
Yeah, no. Like I really like to eat eggplants. So
oh, you just like the thing, right?
Yeah, exactly.
Well, my son is a big eggplant fan as well. But yeah, I have the impression that sometimes these acronyms are made a little bit, once you have the term. So the whole squid thing from our side, I think, didn't also directly originate from the original thing. Yeah, I guess cell to location is a bit more self-evident. Thanks, Vitalii, for keeping things simple there. And then we end by a keynote from sort of, I don't know what happened there, from Johanna Klughammer, from University of Munich here in Germany. We thought we'd bring in more early career PIs into the game. Johanna just graduated from a postdoc position from Avi's lab, works very much on Murfish, and I think it's going to be very exciting to see where she thinks the field will be going. And I think Anna and Giovanni have been asking her to really sort of specifically focus on these things. So I think it's going to be really fun set of events. If you can't join for the whole thing, I believe everything is recorded and you're going to sort of learn about all of these things, but yeah, just try to stay with us. I think now we have sort of more than 600 registrations. Let's see how many people go. We do this asynchronous Q&A and also a demo on both Zoom, but then we also have a Slido for interchange, and then the Zulip stream as well as the GitHub for connection. So I just had to learn about Zulip. I don't know how many of you guys know about that, but I heard it's like the coolest Slack. Is that roughly correct? I didn't quite. No, I didn't quite get together with like the thread handling and so on. So yeah, just gonna learn about it. But I kind of like the loop though. So that's my. But yeah, let's try to see how that works. And we decided to go into the SC-verse one. I'm gonna say in a minute what that is, because we've been realizing there's more and more tools sort of happening in our Python-y type of environment. It's actually a good idea to put these under sort of one hood where we can maybe make sure that we exchange researchers. So we're going to do it via this one. Yeah, please just do this. Maybe someone can put these links again in the chat. Also, I guess we have maybe some Google Docs also where people can look into if they don't sort of directly look at that slide. All right. Yeah, we have a means of asking questions. I guess you know how Zoom works. So just raise your hand, type in, use a chat. I don't think I have to go through this again. So super briefly from my side, something like 10 to 15 minutes where I think the field is currently at and where we're going, what the focus of this thing is. And I promised Anna and the others to be really short about it, so I really try to do that. Just briefly, we're all interested in single-cell biology, right? Actually, I came into the field, I think many of us did, like in the early days, check this guy out. I mean, Alex von Ludner, a true legend, like a physics guy who has been doing essays and doing all these measurements. Many of us just come from the microscope, right? Because I mean, that's obviously how cells have been found, and it's sort of these first potential also multimodal measurements that we had at the time. We had a bunch of fusion proteins. I sort of, for example, looked at time-lapse microscopy in a collaboration with Tim Schröder, who's now at ETH, where we've been trying to understand stem cell decision-making in blood. And yeah, that has been sort of the driving thing for a long time, but of the course, it's kind of targeted, right? So came along this untargeted omics biology of thousands of genes, but then usually ensemble averages. So this is a very nice contrast. Yeah, and this was what Jan-Philipp Juncker and who was at the time a postdoc with Alex have noticed in this beautifully called review, Every Cell is Special. If you haven't read that, just check it out. But then they sort of say, yeah, there's all these techniques came along where we're now in this era of obviously being able to observe many genes and many single cells at the same time. But of course, this comes at a loss, right? So in these cases here, in IH and so on, we always, also in fish, there was always spatial context. This was, of course, lost up there, right? We typically disassociated cells, and it was super successful. It obviously still is in single cell RNA-seq. But now, with spatial transcriptomics and other type of techniques such as targeted spatial proteomics, if you want to call it this way, this has become popular and has been already noted 2 years ago by Nature Methods to be sort of method of the year to be able to do that, and many follow-ups now. And I also just checked. Do you guys know Nature always does these upcoming techniques to watch? And for 2022, they actually put, sort of besides quantum computing and protein folding and AI and so on, they put in spatial multiomics. So this is becoming a thing. Some first papers coming out. So I think it's a good time. This stuff is really timely, and it's important to understand how we deal with these things. Yeah. I'm sure many of you have seen these different techniques for doing this. Is, for example, based on these spots-based types of assays from Joachim's lab, where Alma also has been from, and I think many of us have been seeing the commercialized version, for example, in Visium, potentially higher resolved ones. And then, of course, fish-based assays sort of being on the other side of having much finer granularity. So those, I think, at the moment still sort of in the 10 micrometer range, something like that, going much smaller now with Visium HD, and I think Joachim had already papers on that. Fish-based often being subcellular, even as you can sometimes observe and then sort of see substructures there, and then sort of understand how particular proteins are localized, but then typically being targeted, and there's all kind of things in between, merfish and so on, also being upscaled, commercialized. So I think it's exciting times and very different types of analysis and early pre-processing questions. You might have seen this one paper from Peter Karchenko's lab recently, where just calling these type of protein, these type of fish-based locations were actually realized, I think, with what was the approach? Some type of spot-based Markov field type of localizing, so some Markov jump process type of thing. Really exciting stuff. I don't quite remember, but I thought it was sort of very fancy. It was just going beyond this typically concentration-based things that we usually do. But the eventual goal is that you're able to somehow then again call cell types, as we often do, from disassociate single cell RNA-seq, and then try to see which context they are, learn about cell communication, about locally specific patterns, do differential analysis, all these things that we usually do in RNA-seq, right? So here in this recent review that Giovanni, David, and myself did together with Aviv was where we've been talking about maybe more even spatially resolved omics, right? Because it's these protein-based assays always around as well. So here, this will be for RNA. If we really on the very coarse-grained level, we'd be doing spots. You have then these spot-based assays, but then also laser capture microdissection. Have you seen? I guess we should add that here, Giovanni. This one from what's called deep visual proteomics from Munn Lab recently. Have you seen this one? This actually falls into that category as well, where they use SCM, right, and then sort of do local proteomic resolution, like ultra high throughput, and obviously imaging mass spectrometry from Bodenmiller and other labs. Then single cell resolve techniques. This would be spot-based ones. We spoke about already briefly, stereo-seq and a bit speed, but then also fish-based ones, right? So if you make the spot small, if you really call the fish probes, and of course on the protein side, you could be doing IMC again, spectrometry, or also these labeling things. So essentially all kind of cool variants of immunohistochemistry, co-decisive, the 4i also from Pelkman's lab, popular thing, which is sort of really going the subcellular resolution. So this is sort of the range that you can do, and you can imagine, right? I mean, so far, like in disassociated ones, this is sort of the level. Now, of course, if you add space, you can go very coarse-grained, very fine-grained. And this is all super exciting, in my opinion, for the modeling side of things. We've been like 10 years ago, people be talking about systems biology, about multi-scale models and so on. In this case, this becomes more and more tangible. But let's stay sort of a bit realistic and just get sort of our data organized. And in order to do this, we need data structures. We need to be more hands-on. I think for this, I think making proper tools is important. So this workshop is now not so much about the vision and where things are going, but how can we very concretely analyze the spatial single cell omics data sets that we are more and more facing in our daily business? And of course, how can we also identify potential limitations and things that we still need to do and that are missing there? Here's an example of the different spatial omics data sets that we have. And there's all kinds of tools for processing each of the particular modalities. And I've been trying to sort of enumerate a few. I'm sure there's many others. And there's some toolboxes that put some things together. I believe Starfish is one of them. Wasn't this the CZI project that has actually different type of pre-processing things? But this is not our focus here, right? So we kind of assume that you have been doing already a bit of an effort, depending with a specific tool, that you get this type of resolution of images. Might be not as much fine grained or something like that. Unless you want to do like a spatial analysis, right? So this is sort of where we're going to start. And then maybe the question turns into trying to search for variation. This has been from an older review from Aviv and Nir Yosef. You remember this was sort of when we didn't yet have the proper way of what we want to do with our spatial, our disassociated single cell data sets. And then you've been sort of thinking, yeah, you want to call cell types, you want to look for transitions, for for essentially developmental processes, but then also a little bit space at the time. You're just mapping stuff on landmarks. And then, we extended that we saw we need to include batches and labs because we have variation. So this was sort of the time how we call our analysis, essentially variation analysis. And then recently in this review that I was mentioning before, we've been trying to sort of generalize this type of notion of finding finding components now also in the spatial setting. And this is what we then did. Obviously, we added those 3 dimensions I was just saying about subcellular and the cellular expression. But then you could also call potentially, depending on the technique, maybe morphology, if you have also some type of sort of high resolved microscopy picture of the cells. You can maybe look for covariates, or you would then look really for the big spatial dependencies, such as local cell interactions, but then maybe also modules of tissues. And I think some of these tasks are not as clearly defined, and new methods is coming forward. Once you have this additional spatial dimension, you can then look for processes where things are happening. That could be, for example, a very fine-grained time scale, or also a very, very coarse-grained one, depending on what you observe and depending on the technique. If you have a fish-based one, you can maybe do more than a spot-based one. And then you would be trying to look maybe for interactions to more microenvironment where we see sort of T-cell activity and so on. You could maybe think about the whole trajectory analysis again in a spatial setting. What type of this sort of cell differentiation happens also in a spatially resolved fashion. Export some of these big latent space learning techniques we did in this associated case in this setting. And in case there's a bunch of ideas around, and in that review, we've been trying to categorize those a bit, looking around in literature, and this is sort of also how we came up with this list of tools that we want to present today. And what we think is sort of as most important part to get started, obviously, is image processing, segmentation, registration of the coordinates to make things map onto each other, and then putting things in a data structure. This is sort of absolutely crucial. If you mess up at these things, whatever you do downstream is going to be bad, right? And ideally, I think that's kind of cool. What happened in this associated setting, we've been able to agree on data structures that really made the, I would say, ecosystem of single cell tools so friendly. Has anyone worked before on microscopy in different settings? This is like a mess, right? There's like so many techniques around and tools and there's not even databases where you can download standard resources. So this is kind of nice from the omics community. And with Sura data structures, as well as data in our Python setting, these things are really popular. Once we have that, you might want to, if you have a spot-based techniques, to deconvolution and integrate maybe also this associated setting. There's obviously the cell location others are focused on. Maybe want to do a multimodal analysis if you have also more than just one modality, or just like within different genes, want to see what's shared. You want to check for spatially variable features, either within a particular sample, just which gene is sort of localized, which is kind of all over, or you want to then do this type of thing differential. You want to call cell data neighborhoods. You want to see what's the tissue context looking like. Is it like always a T cell sort of coming up next to a particular tumor cell and there's some interaction happening? This can be formulated in a graph of cell types interacting. We'll talk about that later as well. And then, this is, I don't think there's actually much around yet for that sort of power analysis, unless you correct me there, Giovanni. But this is sort of our outlook thing. Of course, once we have all of this, we really want to go in experimental design. You want to say, hey, for this next experiment, maybe do a targeted thing. Don't do all probes. If you want to do a fish-based ones, maybe just like focus on these few. Maybe you don't need so many individuals, like just stuff that we do also on the single cell associated setting. All right, so this is the overview of stuff that you could be doing. There's actually this nice table in the paper of a bunch of references if you want to read up on that. Also with sort of a bit more fine-grained classification than I was just going through. Check it out. We'll be able to go into details now. But what we want to do today at this workshop is speak about eggplant, putting there really in the sort of in the registration of coordinate systems, like kind of, and also finding a maybe like a more general set of coordinate systems across tools. Hope I'm doing this correct, Alma. Looking forward to be educated in more detail about the eggplant there. About sort of this analysis, both various features, but also neighborhood and multimodal. We've been putting forward the squidpy as sort of a framework to sort of also be modular attached to it and has an accompanying data structure. And we really try to make stuff interoperable here, and these tools already are. We speak about cell to location, which is, I think, a fantastic tool for really doing robust deconvolution. There's others around, and obviously people are using these, and this is all fine. This one has been scoring, I think, really well in recent comparisons. And also sort of plays very nicely with the existing stuff. So I think it's really interesting to check that out. Has been also in our internal benchmarks been doing a really great job and been very useful. And then and this is sort of a tad more speculative. Of course, for cell-cell communication, you can use the self-O2B and similar type of things that you would do in the disassociated setting. So just like go receptor ligand type of thing. But we have this spatial context. We should be doing more with that. And there we've been thinking about using graph convolution networks for doing this. And actually we have some quite nice results, even with sort of very small, very low complexity graph neural networks doing things. And it's not as complicated as sounds. You can potentially call cellular communication. So this is what we try to do. How does this stuff tie together? And this is just a super short plug, and then I'm done with my overview. This is something we call SC-verse. So we've been thinking, we've been coming up with these tools, but like so many others, like how do you create a community around that? Also, so that you can maintain things, that you can actually be like robustly make sure that if you build your stuff on that this is going to be around in 5 years or like at least 3 years. Like how can you get developers power on that? You can't just pay for maintenance all the time. So we need to interact. And this is something we learned from many other communities. And we've been trying to put this together. And I think the core, how many things focus on is data structures. If you have a bunch of data structures in Python, has turned out that many tools use n-data or as extension for the multimodal case now more increasingly view data, right? So what we started with scanpy and data has become like a really nice ecosystem of things interacting. So we've been trying to put this on wider feet, so to speak. So together with the Stiegler lab, the Nir Yosef lab, and many other labs, we've been putting together a bunch of core tools to think this is stuff that should interact and talk with each other. And then we've been trying to build an open ecosystem around it. People can just say, my stuff runs with these tools, and then you sort of get like this benchmark type of stamp, and then we have a bunch of use cases how to really implement these things. And of course, we want to keep stuff interoperable with R and Julia, but this is obviously not focused on the Python side. And for this, we've been really trying to come up with a community, and a lot of thinking of the SC-verse driver people, in particular, thinking about Isaac. Lucas Giovanni, David, many others, in Alex, in this setting, been thinking about like how to talk about these things. Danilo also very much from the Moon side, how to do joint development and really make sure that we have this thing. And I think this particular thing, when you have like this automatic test and make sure that your tool sort of plays nicely with this, is something that we've been trying to come forward with. So we're going to talk more about that, but I just mention it here because you think that these type of spatial tools will be becoming a big part of that thing, because this field is just exploding, and the stuff that we were talking today all sort of plays nicely with these. So I guess we should maybe add eggplant here somewhere, and I guess we have central ligation here somewhere as well. If not, I think that we should potentially add that to that figure to one. Maybe we can make up. Right. So what stuff we want to present today? It's going to be 4 different tools, and maybe I'm just going to skip that. Your scanpy was our disassociated case. People in the lab are funny because they thought this is here a little animal, so they called it squidpy, and yeah, we came up with this thing. I actually don't tell what this is about. I think, Johanna, you're going to make this presentation in a minute, right? Next one will be cell-to location from Vitalii, who's at Sanger and UK, working together with Olli Stiegler and Uma Bayraktar, who's come up with this kind of really nice type of tool to make essentially a single cell RNA reference on top of a spatially spot-based assay with just commonly a mixture NB type of model to call them cell type abundance and localization. Sorry for messing up the scale here. You're going to learn about that in a minute. Alma will be talking about eggplant. Check this out. So obviously it is GP based as I would think, and it's a way to transfer information to essentially this common coordinate framework question, stuff that we had questioned in human cell atlas for a long time. And then also enables, because now it's on the same coordinate system, to actually do differential expression analysis. Because before, if you don't can't stuff can't localize, how would you compare these two things? And last but not least, we talk about learning cell combination from spatial graphs of cells. We essentially build these graphs and call how neighboring cell types interact in such a type of framework. And you're going to learn about that in these next 2 days as well. So this is what we plan. Thank you for joining this workshop, and I look forward to you listening in, the whole discussions. and seeing how this works. And, you know, if these things work out, I think it would be a cool idea to follow up with later such iterations. Thanks. Are there any questions? Sorry, Anna, go ahead.
Yes, let me share my screen, the Slido. So you can either raise questions directly in the chat or using the Slido. I'm sharing here again the code you can use to access and vote questions. Yes, we will share the schedule. We actually shared the schedule already on Zulip, but I will also share it in the chat in a minute. And then we have a question for Fabian. And the question was whether you could elaborate more on what you mean by spatial power analysis.
Thanks for that. So spatial power analysis is, as any power analysis, something that helps us design the next experiment, right? I don't know how you guys do that, but I have the impression in very many cases, we just say, you know, there's, I don't know, 3K euros, let's make this scale of an experiment. And I guess that's kind of fine if you want to be explorative, but if you kind of, for example, if you know something like, you know, this is this level of expression that I would expect, maybe because I have a disassociated type of setting or something differently, you know, then I want to design a particular experiment around that. I just put in the chat an example of how Matthias Heinig lab has been actually proposing to do this with a disassociated setting, quite important, for example, for EQTL and related studies. And I think we should be doing similar things also in that case. Okay. If there are suggestions, be really learn more about that.
Thanks. And then we have another question which isn't directly tailored to you, Fabian, but a request that we should share the Nature Biotech review paper. I think we will do so again in the Zulip. Giovanni will post it there. So we really encourage you to join the Zulip space, as this is the easiest way for us to communicate with all participants. Yes, any more questions in chat, or you can also raise your hand and directly ask Fabian. If not, then I would say thank you, Fabian, for the nice introduction.
Anna, can I just like share one more slide? Because
sure.
I forgot to do that. And I really wanted to get people's question in. Can you guys get active briefly? Do you see my slide here?
Yes, we see it.
So if you could just go to Mentimeter and fill out that slide. Let me just like see if I can maximize it. Do you still see it? Do you see the max now? I think you should, right?
Yeah.
So if you could just go to menti.com, insert this code here, and just let us know what you find important before we bias you, right? Because we're obviously going to learn about these things, and Giovanni says, you know, you need to do that, and then Alma says, you nothing else than Gaussian processes or whatever type of vegetables to do this and that. Let us know what you guys want and what you guys think, maybe also your own research that becomes important. I think it's quite a unique opportunity for us to have 280 people so sharing their brains with us. Okay, everyone type something in, please. Yeah. 3D, thanks. Fantastic, things come in. Really like that. And now this can be self. So if you like some of these terms, just type them in again and add more to them. So I guess we need to group some because cell interaction, if it's wrongly spaced, then there might be sort of similar, but the deconvolution, yeah, I was expecting that for sure. Immunotherapy, okay, some of the applications becoming interesting. I like that a lot. Codex, people, multimodal, trying to aggregate here a little bit. Does it show something? Okay, we have already 38 people entering stuff. So please, everyone who's listening in, just briefly get in there and type in some things that we find important. This is going to be really useful, I think, for us to hear. It's going to be a lot of cell-cell communication. It's kind of what you expect, but if you have a spatial setting, we'll do it. I like that people really stress the 3D part here as well. Okay, people want to talk about algorithms clustering, cancer heterogeneity. Yeah, I get that. Giovanni, Anna, you can type your favorite stuff in there as well if you haven't done so yet. You did already. Appreciate that. Anything else I should highlight? Okay, lung pathology. That's cool. Yeah, I was actually expecting. I mean, I didn't show any examples, but you know, in some of the tissues, this is obviously sort of quite sort of getting much more popular and scalable. I think doing stuff there is going to be exciting. That's interesting. Some people speak about multimodal, and even though I heard it's fancy, I don't know much of big data sets where people really get multimodal. But maybe we could sort of add multimodal disassociated stuff on top of this and do things with that. All right, let's give it like just one more minute. And then take a snapshot of it. Yeah, microenvironment, I think, is an obvious task there. There's people speaking about trajectories as well. Yeah, I've been thinking a bit about that. It's not as super clear what you can do with that, particularly because resolution was still sort of on the spot level in many data sets that we've been locally generating. So it seems as if we are reaching a bit of a steady state here. Thanks for taking the time. Thanks for doing this little experiment with me. I'd sort of add up a bit more, and once this is there, I'm just going to briefly, tweet it or something. But thank you for doing that. Stage is yours, Anna.
Perfect. Then I would like to take the opportunity and announce our next speaker, who will be Giovanni, and he will give an theoretical introduction into the method squidpy and the tool squidpy, and then he will show a live tutorial on an exemplary tutorial. And I would again encourage everyone to join the Zulip, especially if you have any installation issues or if you have questions, join the Zulip and raise it directly to the team, or use the Slido. The link will stay the same and I will post it again into the chat. Then I would say, Giovanni, stage is yours. Yes.
Thank you. Thanks a lot, Anna. Thank you, everybody. Thank you, Fabian. Let's get started with the workshop. So we're going to have squidpy and ncem today, and tomorrow we'll have spatial location and eggplant. I'll start with a couple of slides on squidpy. You can all see the screen, right? Should be fine. And then we'll go straight into the tutorials. So first off, a little bit of motivation. So as Fabian mentioned, there was kind of an emergent ecosystem in Python of tools that tackle different aspects of single-cell data analysis. Scampy may be one of the most popular ones, but then many others have been developed during the years that aim at different aspects and modalities, such as Moon or SCBI tools, Celeron and CirPy, just to mention a few. Now, what all these tools have in common is that they rely on the same data structure, on the same data representation. So, data for, let's say, single modality analysis, and Mood data for multimodal analysis. And this common infrastructure really made the community benefit a lot from interoperability, because it's now relatively straightforward to plug different tools and different steps of the data analysis in the same environment, and therefore benefiting from this interoperability. Now, spatial transcriptomics or spatial molecular data in general started to become more and more important, and more technologies have been were developed. And so this was our motivation to develop squidpy. So with squidpy, we basically tried to create a sort of a bridge between the single-cell analysis ecosystem in Python, together with all these tools and these data representations, as well as frameworks are available in Python that could deal with new aspects, new data representations, data formats, data modalities that these spatial molecular data were bringing, essentially. And these are some of the libraries that we make use of in squidpy that essentially revolves around image analysis and handling and manipulation of large arrays, so oftentimes it's large images. Now, as I mentioned, we kind of got together more formally now, and there is organizations that tries to standardize and maintain all these tools as SCVerse, and squidpy is part of it, but many other tools are part of the ecosystems and really benefit from this type of a shared maintenance and shared data representations. So let's get into squidpy. So when we talk about spatial molecular data or spatialomics data, we actually mean many different technologies, quite diverse as well, right? So some technologies measures gene expressions by sequencing, others by imaging. Some other technologies measures protein expression, for instance, by means of antibodies or mass spectrometry technologies. And so essentially each different technology to acquire the data has its own tools that have been tailored for that specific pre-processing aspects. These are some of the ones that let's say most popular for some of the data sets, such as SciSif, IMC, and Murphy, so fish-based data set and so on and so forth, or Space Ranger for sequencing-based data. And so each of these technologies have their own tools to do the pre-processing. So what we sort of aim for squidpy is to introduce 2 new abstractions on the data representations that these tools produce downstream, that then can be used to do analysis on top of it. So the 2 new data representations we introduced in squidpy are the spatial graph. So it's the nearest neighbor graph based on spatial coordinates, which encodes essentially the spatial information. And then the image container, which is a sort of a container to deal with the large tissue images, which are often acquired in spatial transcriptomics data. And then on top of these representation, on top of these objects, we have tools available to do analysis. And we're mostly interested in analysis that sort of the spatial statistics, for instance, linear interactions, but also image analysis tools such as extracting image features, segmentations, and interactive visualizations. I should probably mention that we put a lot of effort in making the objects and the APIs of squidpy to be easily interfaced with what is already available in Python for doing, for instance, machine learning, for doing, for instance, storing and manipulating large arrays with out-of-core capabilities with Dask, for instance. And so we also have tutorials on it where we show how, for instance, data loaders for PyTorch or TensorFlow can be created on top of the image container to do image analysis, for instance. Now, let's get into the more specifics of these data representations and tools. So on the spatial graph, we put a lot of effort in allowing the user to choose which way to build the nearest neighbor graph based on spatial coordinates. And why is that? Because each technology has its own characteristics, essentially, its own features, right? So let's say all the spatial transcriptomics or previous iterations, essentially, of spatial transcriptomics, such as Visium, or more modern ones, such as StereoSeq, but then again, also Visiums, they lie on a regular grid. And so a nearest neighbor graph on spatial coordinates should account for the grid structure of the observations or measure. Whereas image-based transcriptomics or mass spectrometry rely on maybe more generic types of coordinates, and so other ways to build the spatial graph should be available, especially for the users. And I should also mention that tools then can build on top of this representation, not only within squidpy, but also part of the ecosystem. So Anna and David will present later on the node-centric expression models that make use of the squidpy representations for nearest neighbor graphs on spatial coordinates. Then when this graph is built, however it is for the technology at hand, it can be used to do analysis that aims at extracting spatial patterns. So we kind of classify the two main categories of analysis in terms of continuous and discrete features or annotations. So for continuous feature, we usually talk about genes. So one of the tools that are available is to do spatially variable genes identifications by means of sort of basic like exploratory data spatial statistics such as Morant's I, but also maybe more advanced ones such as SEPA, developed also by Alma. Is an example of Visium where we show 2 spatially variable genes that encompass different clusters of the mouse cortex of the mouse brain, and also in terms of ligand receptor interactions, again based on genes. So in this case, we re-implemented CellConDB, a popular tool for ligand receptor interactions in Python, and we not only made it faster, but also we made it use a larger collection of annotations, because these tools rely on a annotation databases on ligand receptor interactions. And so now it's possible to parse essentially the OmniPath, which is a large database that aggregates different ligand receptor interaction databases. And actually we're going to use that during one of the tutorials. And then in terms of discrete features, so we can we provide again exploratory data analysis tools for identifying interactions between clusters, for instance. So we have examples on the neighborhood enrichment analysis. This is an example on the SecFish dataset of the mouse gastrulation, of the mouse embryo, sorry. And this analysis essentially aims at quantifying co-occurrences in space, neighbors in space of different clusters. And we show that the enriched clusters interactions are also found by the original publications. Other ones are co-occurrences, for instance, the co-occurrence scores, where we essentially look at whether, given the observation of one specific cell type of one clusters, what are the other cell types that are co-occur, that are observed together at increasing distances. So again, we have the expression of the mouse brain from Visium, and we find that condition on the hippocampus, the pyramidal layers, the orange and the green ones, co-occur at close distances with the hippocampus. Now, this is not this is sort of like a validation, but provides you with a hint of what this type of analysis can be used for in your data. And then again, we also introduced the image containers, which is really a wrapper of X-array. So it's a wrapper of essentially storage of large arrays, and we provide it with a bunch of methods that performs analysis on this image. Now, in this case, for the image container, we're mostly targeting spatial transcriptomics data. So for the case where, for instance, for segfish or mass spectrometry, maybe they're not the most suitable, maybe it's not the most suitable tools for doing the image analysis, as there are other ways to do it, but it could, of course, be tried out. But anyway, in the case of Visium, we really thought that for Visium and spatial transcriptomics data in general, where we have sequencing-based spatial transcriptomics, the image, which is often acquired alongside the experiments, should be treated as a modality, right? So it should be featureized, it should be used for potentially extracting morphology and so on and so forth, but also segmentations of nuclei, if it's possible to segment the nuclei on H&E or fluorescent images. And then we provide interactive visualizations with Napari. So in the tutorials, we have examples on actually Mibitov preprocessing, so mass spec data. Also, how can we plug different segmentation models, so pre-trained deep learning models, into the image containers for doing the segmentations. And this is particularly useful because, again, of course, this analysis, the segmentation can be done with sort of standard image analysis pipelines using Watershed. But if in case there is pre-trained models for the data at hand that can be used for segmentation, it's really easy to essentially plug it and use it to segment the nuclei or cells of the image in the image container. And then we have a visualization with Napari. This is something that Napari is a very popular tool in image analysis for doing interactive visualizations, but also really analysis. There are a lot of widgets that allow us to do analysis. And we developed a wrapper, essentially a plugin, an Napari plugin, to explore interactively the gene expressions, the annotations stored in data together with the tissue. So you can see here, for instance, we're plotting gene expression information on top again of the mouse brain from the next genomics Visium. And you can change how different layers are visualized, overlaid, and so on and so forth. You can also actually perform some type of analysis, such as annotating tissues. Annotating regions, in tissue. You can essentially draw shapes around regions of interest of the tissue and then export them directly on your data in the same session, the same Jupyter session. So with Shift E, you export information, which is stored as a categorical series as a Boolean series in undata, in the undata ops slot. And it's all part of the same Jupyter session, so you can essentially use this for further downstream analysis. This is particularly useful because you don't have to leave the Jupyter session that you're running the analysis on. So since publication, we released a couple of new features, and I'm going to briefly mention what are they about. I might show some of these things, but not all of them would be part of these tutorials. But we have examples and tutorials in the documentation online. So the main feature has been to refactor static plotting functionality in squidpy. So before we were using the one in scanpy, and now we have a much more complex and rich static plotting functionality directly in squidpy, which also allows for segmentation masks. I'm going to show examples in a second. We made a function to download conveniently many transgenomics data sets. That's particularly useful, for instance, in case of method development for publicly available data sets from transgenomics. we now start to support the cases where multiple slides are present in one data. So in the same way as in single cell, if you have different data sets, you store them in one data file, and you will just treat the different data sets as a batch, essentially, right, with a batch key on your data ops. For spatial data, it's a little bit more tricky, especially if you look into if you're looking into doing analysis in terms of spatial patterns. So each coordinate system of each slide is independent, and so the way to build spatial graph and the analysis to do analysis on top of the spatial graph should be should essentially be aware of this. And then we also have reading functions for new technologies, not only vision but also vision and nanostring. So this is an example actually on nanostring data set. So these are available publicly available. This is from nanostring. It's, I believe, it's a section of the different sections of the lung, and in this case we are showing the functionality of the new spatial segment plotting. We are plotting a specific marker. In this case, fluorescent markers already provided by nanostring, and we can look at it really across different resolutions, right? So we can look at the whole data set. So in this case, we have 26 slides, and we can plot them all together with essentially one line of code. We can also select specific slides that we want to look into it. Again, these are just the top two, I believe. But then we can also segment, we can also sort of zoom in and crop specific slides for looking at like very fine-grained details. We can also plot the segmentation mask with this erosion effect so that you only see the boundaries of the segmentation masks and not, and as well as the underlying image. And again, this is just to show more of the segmentation plotting, but also the scatter plots, the circle plots in squidpy, which we're going to be using today. And then a little bit on the future. So what we are working on right now, we realized that maybe one of the major bottlenecks for both storage, data manipulations, and methods that could be built on top of this, we really need new data formats and new data representations. So we're working with a bunch of people on essentially integrating what we have in Undata with what the image analysis ecosystem uses, or it's starting using more and more, which is the OME and GFF format, to essentially provide a new spatial data format that is can be used in the ecosystem, in the Sessiverse ecosystem, and really makes use of the latest or the sort of the cutting-edge technologies that Python provide for working with large data sets, also in the cloud. More on data representations for storing transcript locations with awkward arrays, and then again the workshop that we're going to talk about today. So with this, I finished the presentations. I'm gonna exit. I'm thinking maybe it's I could take a look at Slido really quick and see whether there are any questions. Maybe we can do a couple of minutes so people could ask questions, and then I will jump right into the tutorials.
Yes, Giovanni, we don't have currently a question on Slido, but we had one on Zulip asking what would be the easiest starting point for a person coming from the omics field to start with spatial transcriptomics. Also to get your opinion on this.
Yeah, this is a great question. So coming from spatial transcriptomics, I think squidpy is a good entry point, I'd say, because it really uses a lot of what scanpy and SCDA tools and so on and so forth uses, especially Undata, right? And also the API, so the way you sort of calls methods on Undata, it's very similar. And I would say maybe any of these tools that we are going to cover today, ncem and SL location and eggplant, are good starting points. I think maybe a comment is on the type of data that you're generating or that you have to analyze, because in the case of sequencing-based methods such as Visium, maybe DBSeq, for instance, and so on, it's kind of relatively straightforward because they're still sequencing-based, so you don't really need to do a lot of image pre-processing. For other tools, for other technologies, also for instance MassSpec and so on, there are specific tools that are developed for doing the pre-processing that should be looked into essentially, and it's something that we do not really provide or cover in squidpy or in the other tools that we're going to talk about.
Perfect, thanks. Any more questions before we go into the tutorial session? If not, you will also have time afterwards in the Q&A session.
Yeah, I'm seeing on Zulip some questions on installations on M1.
Yeah,
so
it was to sell to location, I think.
Yeah. And on Slido, I still don't see anything. So will these slides be shared? Yes, yes, we can share the slides for sure. Yeah. You can still see the screen, right? Now the tutorials.
Yes.
Okay. So I'm thinking maybe for the sake of time, I can start with tutorials, but please interrupt me anytime for questions that you might have. You can again post them on Slido. I will keep Slido open here so to have a look. Or on Zulip, maybe Anna, you could ping me if there are questions on Zulip, so I could answer them right away. I mean, I guess it would be maybe nice for everybody to have it a bit interactive. If possible, so feel free to really ask questions as we go along. Okay, so I'm gonna start with the tutorials that we have in GitHub. So we're gonna cover 2 tutorials today. One is a bit of an introduction of how Undata stores spatial data and spatial information. And it's useful if you're a bit familiar with Undata, ready to essentially get a sense of where things goes in Undata. And also in case you have data set that we don't have readers for, right? So if you have a specific technology for which we don't have a reader for the data, you can essentially build the Undata following these instructions. And then the second one, we will go through our tutorials on Visium, and I'm going to show some of the functionalities that we have in squidpy for Visium data. I think many of them applies to other technologies. And again, I invite you to look at the docs for how these specific analysis tools can be used for different technologies. So I will start with the first one. So I'm going to start with importing a bunch of libraries. So Undata, scanpy, squidpy, and matplotlib. And now it's taking a little bit of time. Yeah. Okay, so we start with really sort of like the basics. We're going to create dummy data, and we're going to build the Undata on top of with this data set essentially. So we're going to create some counts. So our feature matrix, so our cell by gene matrix. We're going to create some coordinates. So our spatial coordinates, right? This is going to be a cells by X and Y array. And then we're going to create an image. So this is the command. And then we're going to build the Undata on top of it. So the Undata takes the first entry, takes the feature matrix. And then it saves, it's important that you save the spatial coordinates under the ncem slot, the observation matrices slot, under the key spatial. And then we're going to do some basics analysis just to kind of show that this is maybe one of the first things that you would do in scanpy data analysis for processing this data, but it's not necessary. It's just for purpose of the tutorial. Okay, so now we can look at this as it look like. So in this case, I'm using the first iteration of scanpy spatial plotting. Now we have one in squidpy. I'm going to show you later on, but just to show you that essentially it works the same. So both spatial in scanpy and squidpy makes use of the spatial basis, the information stored here. So in opsem, in Undata, we store essentially the data representations, so low dimensional representations such as PCA or UMAP, but also the spatial coordinates. And this is what is using these functions in the back, underneath essentially. We can compute spatial neighbors. Now, one way to do it, this is the function we have in squidpy, and if you essentially look at the doc strings, the signatures, you can see that there are how different ways essentially are, how different spatial neighbors can be computed parsing the given arguments. So I guess the most important ones is the coordinate type, which can be either generic or a grid. And so a grid is useful, for instance, for, again, as I mentioned, Visium or first iteration of transcriptomics or DB-seq and so on and so forth. Generic ones are more, let's say, standard nearest neighbors algorithms on, in this case, a point cloud in 2D. And it defaults basically by grid neighbor 6 because it defaults on Visium, but yeah. You can also select the number of neighbors you want to choose. This is both for generic or grids. You can have different grids essentially. different neighbors on the grid, right? So if you have a square grid, you would have 8 neighbors for a 6. Otherwise, example, you would have 6. But also you can select other ones to increase the radius. So the radius is essentially essentially computes neighbors based on a distance, on a maximum distance for each point. This is what we're going to be using here. So now with radius 3, in whatever the point cloud is in this case, these are defines the neighbors, and we plot a spatial graph. We plot the nearest neighbor graph, the edges of the nearest neighbor graph. Okay. So how do we do that in squidpy? The function, by the way, I'm going to do some modifications here. I have some exercises also in the next tutorial. So I'm just going to run them now, and I'm going to push the sort of solved exercises and the modifications that we do on the way to the repo, so that you will have access to this as well. So the spatial plotting in squidpy for this specific data is called spatial scatter. If we have segmentation mask, we call it spatial segment. And it kind of follows the same signature as the scampy ones with a few modifications. So in this case, for instance, the color is the same. So what we know color on is the same. We need to pass a new argument called shape, because we're now allowed to plot different polygons. We plot circles for Visium. We plot squares for DivXig. You can plot even hexagons. I don't think there is any technology that actually measures hexagon, but it doesn't matter. If you do not have information on the spot diameter, on the observation diameter or the observation radius, whatever. So you don't have like the actual measurements of how big the observation is in pixels in the image coordinates. Then you would pass shape equals to none, and then it defaults to a scatter plot. Now, why is this important? Because in Visium we do have the spot diameter, right? So what we do is plotting the actual circle that encompasses the capture area of the Visium spot. And this allows us to be sort of faithful in terms of representations as you crop, as you zoom in, and so on and so forth. And so it's useful for doing that. In this case, this is just like dummy data, right? So we can just pass shape equals to none. We also have a different key now for the connectivities. So here we would pass the spatial neighbors. Now we pass the spatial connectivities. Why is that? We simplify it because the spatial connectivities are stored into undata.ops together with whatever other connectivities that you have computed, right? So when you call, for instance, scampy.pp.neighbors, it computes the k nearest neighbor graph based on PCA or the feature space that you have, and it saves it in ops. It saves the distances and the connectivities. And this is also true for the spatial neighbors. Now it's a different type of adjacency matrix, because the neighbors now are not based on whatever similarity on the feature space you computed, but on the spatial coordinates similarity, so on how close they are in spatial coordinates. And they are stored under the spatial connectivities. And then we have also the edges width, the same as before. And we can, so we serve the same. I mean, we can change the size to have it bigger and so on, but this is just like aesthetics choices. Now it throws a warning which says, please specify a valid library ID or set it permanently. And this is a warning specifically for cases where you have multiple slides. And I'm going to show later on some examples. In this case, it's useful to have a library ID specified also as also in undata.ops, because then you can plot different slides of the same data sets together and coloring them differently and cropping them differently and so on and so forth. And this is something that was not possible to be done with the first iteration of scampy. Okay, so this is it for the spatial neighbors graph and where it is stored in undata and how it can be visualized. Now let's take a look at the image. Now this is the sort of dummy image that we created. And now it's a bit of important of the metadata that we have with this image. So everything related to the image itself and the metadata is stored in undata unds. This is the unstructured slot of undata. And specifically, it's under undata.unds spatial and then the library ID. This is what I mentioned the library ID before, because if you have multiple data sets, then you would have multiple library IDs, and they will be essentially different entries of this dictionary, this tree. And library ID is always a unique key, of course, for each data set. Then under this specific directory, we have additional information, different metadata. So the important ones to consider are the images, where we actually store the images. And so this is an example. So here we have a library. I just called it tissue42, and we are gonna have two additional dictionaries. One is the images, where we store the images at different resolutions, and one is the scale factors, where we store all the metadata related to the scaling factors of the coordinates with respect to the image. Now, for the images, I only have one image, and I call it high-res, and it can be multiple ones. We can also have segmentation mask, for instance, stored here. And for the metadata, for the scale factors, I store, most important ones I store are the high-ref, the scaling factors. So these are scaling factors of the coordinates. I'm going to show later on what does it mean. And then, as I mentioned before, the spot diameters. This is essentially information that is used for plotting the diameter on essentially on top of the image, to plot, sorry, the spot with the exact diameters on top of the image. And okay, we can just run it. This is how it looks like. Now, it looks like it's the same image. It looks like it's a scatter plot, but it's not really a scatter plot, because we're not plotting. We're plotting circles, essentially. We're plotting polygons, and this could be also the same for square. Actually, I could just maybe try square. You see, we're plotting squares now. Now, the squares takes the diagonal as the same size as the diameters. You can change these information in the signatures. But just to show you a polygon, you can also plot a hexagon. X, I think it's called. Yeah, you see these little hexagons. We can maybe we could have an outline, for instance. Yeah, so we can plot an outline to maybe make them look better. This is something that's also possible for the human, for instance, squidpy, with the same arguments, the same API, essentially. So I guess one way to understand how these metadata interacts with each other is to sort of try different things, right? To just basically fiddle around a bit. One way to do it is to change the values. Let's say now we fix the spot diameters full res. So the diameter is the same as before, but now the scaling factor of the coordinates with respect to the image is half of it. Okay, so if we plot it, you can now notice that the data is looks like smaller, and the image is the same as before. And also, I would say maybe like shifted. You see that before they were like encompassing the whole image, whereas now they are all on the top left. Now this is because now the coordinate system is half as the pixel resolution. And so that's the shifting that's also introduced by the plotting. Again, this is important as soon as you start having to deal with different resolutions of the images or different transformations of maybe cropping and so on and so forth of the spots or of the image. And this is how they interact. So I guess, again, just a way to try out is to change parameters and see how they change. We can, of course, also look at the specific image. We can change the aesthetics of the image on the underlying image. So now we are plotting it in grayscale. Let me check actually, because I don't remember. Did they call it image? Oops. Yeah, so we also have channels actually. So we can also pass image channel, and we can, for instance, look at only one channel. I don't think it's going to change in terms of, oh yeah. So we can plot single channels of the image. I mean, again, this is just an example, but I guess with real data would look maybe more striking. So you can, for instance, just plot the DAPI if you have the DAPI, or specific properties if you have specific properties. This is possible. This is available, for instance, in taxonomic Visium. Often it's possible to acquire fluorescent images, so not only H&E, and then maybe this is useful for this purpose. And then the image container. Now, the image container is a separate thing. It's a separate object. Why is that? Because it stores the large microscope images it's taken, in this case for Visium, for instance. And we have a separate object, and we essentially interact with the separate object and data via the image container. In this case, now we just essentially assign it and show it. So there is not much that it can be done on these sort of dummy examples. It's going to be more interesting on the next tutorials to see how do they interact. Now, it's maybe for you to understand essentially is an XRA data set. So now we have image container, or better, sorry, if I do, yeah, you see the doc string. So the docs, the visualization here, the print is just an image container, but it's really an XRA data set. And then you can look at specific image, and then you get an X data array. So you have the different coordinates, right? So you have the YX, the height and the width, and then you have channels. We have 3 channels here, and then we have a Z. And the Z is because we can have segmentation masks. We can have we can stack essentially different representations of the data, and we would have essentially different as a Z stacks of images. So yeah. Okay, so I'm done with this tutorial. I think I'm on time. Let me check if there are questions here. There is one on Slido. Is there a specific reason for the order running first normalized total followed by log 1p instead of the other way around? Yeah, so this is maybe more like normalization questions on ScanPy, but normalized total essentially performs a scaling by the size factor, by the total library size, and it's usually run before the actual transformations, the various stabilized transformation, which is in this case the log 1p. So you would have to do it before. Yeah. And by the way, this is done because it's counts data, right? Sequencing. So we do this, but in case you have maybe, I don't know, intensities, proteins, so on and so forth, you would choose something else. And maybe just scaling, you know, like, or minimal scaling and so on. Is it possible to do multi-image data integration? Wait, can I show the, maybe I can just show the slide over here, because then you guys look at what image, what questions I'm answering. Is it possible to do multi-image data integration and do normalization? I would say partially yes, but you would have to store different image containers. So we do not really have a way. This is one of the major things we're working on right now. We don't really have a way to store multiple images that have different shapes essentially inside the same image container. So it is possible. You have to have different objects essentially and then bringing them along in your analysis. It's not very convenient, but it would be possible. Normalization is possible to do any type of cyclic image normalization, so blurring and so on and so forth, for the images, for the pixel I'm talking about. Second question, for image channels, are you selecting just by channel index? How would you call channel by, say, a method of proteins? Yeah, now, well, okay, so for the image container, you can select by the channels actually. No, sorry, I misunderstood. No, you can only select by index because we just store the array essentially. For IMC, which is your use case, I think this is a very interesting use case, and it would be interesting to do it. And this is something that, again, we are working towards making a new, let's say, object, which is it's also a neat hybrid way between the image container and the data. And then these kind of things will be possible. It will be more straightforward. Also, I should mention that I would only store like small, low resolution, small images in the data, and I would store anything else in the image containers for larger ones. But unfortunately, this type of analysis is not very convenient right now, but it's something that we're working on. Will it possible to use 3D data image with just a spot coordinates like that? Yes and no. If you mention about different stacks, it is possible. If you mention about real 3D data, like volumetric data, then it's not possible. It is something that we also like to try out. I must say that there's, I haven't seen much 3D data with like enough depth and enough multiplexing, but it's something that for sure is coming. So yeah. One thing though I should mention is that this is on the image side, right? But if you look at the 3D data as essentially a point cloud in 3D, where you have features for each node, for each observations, then you can build a spatial graph based on the 3D coordinate space and do all these analyses as before. So a lot of this is already, you can do it already as long as you don't look at the actual pixel level representation of the image, but you look at the featurized level representation of the image. So where you have already segmented cells, you have features, and you have the coordinates of the centers, for instance, and so on and so forth. I hope it answered the question. I know that both are required, normalization, log transformation. The question, why not log transformation first? Yeah, I I'm not sure how to answer this. Maybe someone else can jump in here, or maybe something that could be useful to ask in the discourse of SCVerse. It's more on the data normalization side, which I don't want to like say things that I'm not super comfortable about. I see David is showing up maybe as a no, okay, yeah. So I think this is an interesting question we could discuss maybe on Zulip and so on, and maybe more experienced people can do it. Are all images stored in their own image container? The images, yes. The data, you can store one data with all the images. So for the large images of the image container, you'd have to store different image containers unless they have the same shape, but this is rarely the case, right? But if they have the same shape, you can store one. You can store one image container with multiple layers, with multiple images, image 1, image 2, image 3. For otherwise, you have to have single image containers. In the data, though, it would be possible. Okay, so I want to move on the tutorials now.
One additional question in the chat, Giovanni. Maybe Robert Sellers wants to address it himself. Otherwise, I can also read it.
Yeah, I see that.
Okay, I'll just read it then. So in terms of the space ranger outputs, the high resolution image supplied is actually quite low resolution. Can you also import the original full resolution TIFF supplied to this to space ranger for image analysis? Yes. Yeah. Sorry, that's a good point. When I mentioned the high resolution image, I mentioned the TIFF file that you use as input for space ranger. Now, the output of space rangers contain two different resolutions, the high res and the low res, but these are both PNG images, which are only useful for visualization, but not for analysis. So I agree. For the image container, you should store the TIFF image that you use as input for space ranger. Yes, because that's really the high resolution that we can do segmentation and so on and so forth. Awesome. Okay, so I'm going to move on the next tutorial. Now we look at some analysis. So I'm gonna briefly reactivate this. Okay. So now we open we load a new dataset, the Visio HD dataset, and in this case I'm passing to the image container the high resolution, the output of space ranger in high resolution, so not the original TIFF image for the image container here. And this is just again for the purpose of the tutorials, but you should store the TIFF image, and you can load it. You can load the TIFF image. You can just pass the path of the TIFF image, and you can load it. Does it make sense? Yes. So another point is that I need to pass also the scale factor here, because the pixel coordinates. So what we store in data.ops_m spatial. So this information here is the units is essentially the same as the large microscopy image that is used by space ranger as input, but the one that we are passing here to create an image container is the lower resolution, so the high res version of the output. And now we need to pass the scale factors, otherwise it will not work. If you have to pass the TIFF image here, so you pass a path to the TIFF image, then you don't need to pass the scale factor. Okay. Okay, so let's look at the image features. So as I mentioned before, one thing that we can do in squidpy is to featureize the image, meaning extracting, for instance, summary statistics or more complex morphology information from the underlying image, and then essentially you really use it as a different representation of the data. So what I'm doing here is computing these image features on the image. The feature class is called summary. So these are the summary statistics, so the mean, intensity, the challenge, and so on and so forth. And the feature name is feature summary, and then the scale. Why I'm doing this? Because I'm computing them for two different scales, for the actual spot size, and then for double the spot size. And this is basically to get context, right? To get essentially computing the features of the image on a larger area. So not the actual spot in pixel resolution, but like larger ones. And what I'm doing then is I'm combining them and concatenating them and assigning them to the ops_m representations. And then I'm going to pass also the columns essentially. Now, this kind of analysis could be done with mu data. So in mu data, you would store the one data which has the gene expression information and one data which has the image level information. And you would build a mu data
from it essentially, and then you just pass different representations on the analysis downstream. Now, in this case, we didn't really have, when we developed, we didn't really add mu data. So this was 1 year ago. So now we have mu data, so we could include mu data. We don't include in the dependency of squidpy because it's something that user maybe should want to do it for themselves, but it's possible to be done actually. And there is some of the analysis I'm going to show you downstream now. It's something that you could, for instance, do with mu data as well, and mu data and muon, I would say actually. So this is doing the computation now. And now we have an helper function that essentially extracts the features saved in undata.opsend. So well, let me show them really quick. So this is what we computed. So this is a concatenated data frames for the 2 scales. And then we have the summary statistics of the features, like the quantile, the standard deviation, the mean, and so forth, for 1 scale and for another scale. So if we don't have an index, it would be the first scale, otherwise we would have the second scale. Okay. And this is a pandas data frame, which is stored on data offset. And it has shape. This is the number of spots, and then the total number of features in computing. I invite you to go to the tutorial, to the documentations for different type of features. So you can also use a lot of acyclic image, region props kind of features, right? Morphology and so on and so forth on the image. What we do now is that we extract these informations from the undata, and we create a new undata, which no, sorry. In this case, we extract this information and we compute Leiden on this representation, right? So what we do is that we take the undata.opsm, so the features, which we pass to this helper function. We create an undata with these features. We do scaling, PCA, neighbors, and Leiden, and we essentially get a cluster informations on the similarities of the image features. So not on the gene expression, but on the image features. We assign it as a new slot in data.ops, and then we plot it. Okay, so this is how the clusters would look like on the image features. So the same type of Leiden clustering that you would do in scampy compared to the clusters in gene expression. You can see that they're way more, I would say, maybe noisy, more scattered. Although some of them seem to resemble it. So for instance, for the fiber tract here, they seem to kind of recapitulate more or less also at the morphology level some type of similarities. So this is, of course, this can be done maybe with richer representation, maybe with fluorescence and so on and so forth. And what we're doing now really is basically looking at the clustering on 2 different representations of the same data, right? One is gene expression and one is image features. And again, by image feature, I mean the summary statistics on the channels, which are maybe not super interesting, but some type of indication essentially. Another thing we can do is to do joint clustering. So we can cluster with Leiden on both the adjacency, so on the graphs, nearest neighbor graph, both in terms of gene expression features or image features, or gene expression features and spatial features, as spatial neighbors. So this is the exercise. Okay, well, I'm just going to run it now. So this is the kind of data you need to do. So we would have a new undata. We create undata image, which we essentially recompute from before. We compute neighbors on the features of the image features, and then we extract the adjacency matrix here, undata image opposite connectivity, and we sum it with the adjacency matrix from the gene expression connectivities computed before, and then we run Leiden on this joint adjacency matrix. Okay, and we call it the joint Leiden. So I'm just going to run this. And now I can plot it. And this is how it will look like. So now the clustering on the left is a clustering performed on essentially the joint representations of image and gene expression similarity. And this is essentially how I would compare with the gene expression clustering. Now, the same thing can be done, of course, with spatial connectivities, right? So if we now, instead of using the image feature, we use the spatial connectivities, So did we compute them before? I don't know, actually. Let me check. So in undata.xp, I do not have them yet, so I'm going to have to compute them. And now this defaults on Visium, so I don't have to pass any argument because we have Visium now. And now we do have the spatial connectivities. So what we do now is creating this joint adjacency graph. This is a joint adjacency matrix. This joint graph with spatial connectivities and gene expression similarities connectivities. And we call it joint Leiden graph. And then we plot it again. And this is the result. Now you can notice that it kind of look like we are sort of creating like a tessellation of the tissue because we have the same weights between the gene expression and the graph, the adjacency graph. I guess this analysis could be done, for instance, by downweighting the importance of the spatial graph, for instance, by just a factor like this. And what we are doing now, is a little trick that I forgot. So I need to remove the coloring completely before. Oops. Yeah, this is like the live, yeah, the live demo. So what I'm doing is that it just throws an error because I would have to pass a color map. I don't want to pass a color. I want Scamper to compute it for me. And this is how it would look like. So now what we're essentially doing here is to finding a balance between the Leiden clustering computing on the graph representations and the gene expression representations, the spatial graph and gene expressions jointly. Now, this analysis is something that you can do in one line of code, and there are maybe better methods to do this kind of spatially aware clustering, but it was kind of interesting to see that it was used, for instance, on a paper recently. They published these massive data sets of stereo-seq, and they found these spatial clusters, they call them, I think. So if I just search for squidpy, the spatially constrained clustering, and what they did is essentially what I just showed you. They combine, they compute a graph, a combined graph based on the spatial graph and the gene expressions. They use squidpy graph, nearest neighbor graph, standard one essentially. And then they run Leiden clustering on top to find this clustering showed, I think, in the main figure. And in this case, they don't look like so sort of smooth out because probably the data, first off, is much higher resolution data. Maybe there is more spatial structures, but this is just just to show that this approach doesn't have to work everywhere, but maybe sometimes works. And maybe there are also other methods actually that do the same thing. That sort of try to tackle the same problem. Okay, so I'm gonna go on and show some more other questions. I don't know. I understand that now I'm going a bit too slow. Should I look at Slido? Yeah, exactly. This is let's see. This is exactly what I mentioned. So we don't have a function for that, but you can clearly see that it takes 2 lines of code to do it essentially. Some specific features, yeah, they just to take intensity, yeah. So they essentially the pixel level, the scalar of the pixel essentially. What actually are image features? These image features, so there are different ways to compute image features. In this case, we are computing the summary statistics, so the mean intensity, the standard deviation, and so on and so forth, on the pixels essentially, on the spot essentially, the pixel level representations of the image. For HNE, again, this is one way to do it. There are possibly other ways to do it. Segmentation, for instance, could be performed. Then you could count the number of nuclei you have under each spot. And we have actually tutorials for that on the squidpy docs. In case of fluorescent images, maybe you can have richer informations for that. But this is one way to do it that we want to show essentially. As far as combining these, is there a good heuristic for figuring out the weighting if not being? Yeah, that's a good question. In terms of heuristics, it's a bit hard, I think. So I guess one way is that if you have a ground truth clustering for the same tissue, then you will compute, for instance, the clustering, some type of clustering metrics like NMI and so on and so forth, find a good heuristic for scaling the graphs, and then propagate it on the rest of the data set. But this, I understand, is not oftentimes not the case in the real world. So I think it's a bit of a trial session. It's difficult to say yet. Yeah, I think I answered all the Slido questions. I'm going to go on. There is a new one. How do we validate if the image-based classes are biologically or not? Yeah, how do we do that? That's a good question. So in the squidpy paper, for instance, we show that if, okay, so the summary statistics in this case, in the H&E are a bit difficult to interpret, although they kind of give you a sense of like how much nuclei they are, more or less. They kind of correlate. If you want to go a bit more like in deep, you could segment, the image. Then you could count how many nuclei there are on each spot. And then you could find, for instance, what we show in the paper, that you could define clusters for the cortex, for instance, based on the nuclei density. And so the cortex, the outer layers, they have less nuclei. They have lesser density of nuclei than the one and the inner ones. And that is something that is biologically useful, for instance. But otherwise, it really depends on the tissue that you're looking at and the type of questions that you want to answer. So I hope it's a good. I understand it's like I'm not giving like clear indications, but yeah, I hope it's a good answer nonetheless. Okay, so
Giovanni, we have one additional question in the chat on: do you have similar examples like in the paper where custom feature extractors are used?
Yes. So I'm gonna go to the docs now. So no, wait. You can pass the feature custom essentially. So feature custom essentially, then you would have to pass a callable, so a function that does something. Basically, what this method is doing is that it's giving you the array, the crop array, the crop image under each spot, and then you can use it to do things. So you could have custom ways to do the feature extraction. Another way to do it is, for instance, to do segmentation. So here is what we show here, for instance, in the is it here? I don't remember now. Oh no, maybe it's in tutorials, externals. Yes, so for cell posts, for instance. H&E, let's look cell post H&E. This is the callable that we define. So this is the function we define. And what it's doing essentially is taking the image as input, and it returns also an image as input. You would, in this case, use it, pass it as a method here, you see. Method is just the function call, essentially. You can do the same for the calculate image features with a custom functions, essentially. So in this case, we're doing it for segmentations, because now it returns basically the segmentation masks. So it's not a height, width, and channel, but it's a height and width image only with integers values for the segmentation masks ID. But you could do the same for the calculate image features. And I really invite you to look at the docs, because we have a lot of examples on other ways to do it. Features, texture, histograms, features on the segmentation masks also, for instance. So if you have a segmentation masks available, I think actually we have an examples, sort of like a dummy examples on how to do it. Actually, we do for sure, I remember. Extract custom features, look at that. So now it's a dummy example. I think what we're doing is just we are computing the mean of the array. But this is how you would pass it, basically. You would compute features equals to custom, and then you would have the keyword arguments with the function itself. These are the top. Does it make sense? I hope it makes sense. Okay. So I understand I'm going to go on with the bit with the other analysis. So okay, I'm going to close Slido now. I'm going to answer them later. So now let's look at some spatial statistics. So descriptive analysis for exploratory analysis for extracting spatial patterns in the tissue. So one function we provide is the neighborhood enrichment analysis. This is a bit of an explanation. It's an enrichment score based on spatial proximity. If a spot belonging to two different classes are often close to each other, then they will have a high score. Otherwise, they would have a lower score, and we would call them enriched or depleted. The score is a z-score performed on a permutation-based test. So you have a z-score and a p-value, and you can decide how many permutations you want to do. So I believe that the argument is number of permutations, and the default is 1,000. So what we do is we essentially shuffle the graph, and we compute again the neighbors count for 1,000 different shuffling, right? So that's our null distributions, our permutation-based test, and then we put a z-score on that. So we run it, and we plot the results. And it takes a little bit because we are using Numba, and Numba needs to JIT, so it's very fast as soon as it runs, but it takes a little bit to JIT the function. And this is the plotting. So it's the plotting. Oftentimes, the plotting function is the same. There's the same name as the spatial statistics functions, but it's running a different module. It's in the plotting module. So now we're plotting the cluster because as we computed before, and we can choose a different CMAP column, and that's the result. So in this case, we find, for instance, that the pyramidal layers that are close to each other are enriched. Again, this is not surprising, right? Because the mouse brain has been described very well, but on new systems, on new data, this is interesting nonetheless for doing this type of spatial statistics, for finding essentially communities and so on and so forth. And these are like sort of simple ways to do it. For a more advanced way, Anna will talk a lot about the kind of functions they have in ncem for doing this. Another spatial statistics is the co-occurrence. This does not operate on the connectivity matrix per se, but on the increasing distances essentially. And it computes the conditional probability of observing any clusters given one, given the sort of the condition. And this is the formula. And again, the score is computed across increasing radii size. So it's increasing distances essentially from each observations of the classes they're looking at. Again, you can parallelize it with njobs, and you can also decide the intervals, so how many bins you want to compute this, right? So I don't know, from maximum value, it computes the maximum values per se, the maximum distance you want to look at, but you can also pass it, you can pass bins, you can pass the maximum value, and so on and so forth. The default is just, I think, computing the maximum. I think there is like an heuristic on computing the maximum, but yeah. And you can also parallelize it. So now we run it. In this case, we are computing it on all the conditional probabilities of each cluster against each other, but then we plot specific ones. So in this case, we look at the hippocampus, and this is the same figures I showed you before. This is the increasing distances. Now the unit here is in pixels, in the pixels space, and we see that the increasing distances, the pyramidal layers co-occur with the hippocampus because they're close to each other. Again, not super interesting, it's more for as a validation, but in your raw data, this could be potentially useful. We also have a receptor interaction analysis. So this is CellPhoneDB. I'm not going to talk much about it, but essentially, given a set of annotations of the big receptors, we look at the mean expressions between different clusters. It does not use the spatial information at all. It just computes it as in single cell. Actually, we were aware that many people use this just for single cell because they can run it in the notebook without having to do the command line. It's also pretty fast, and again, the annotations it's larger. But you could come up with ideas to include the spatial information. So, for instance, in the latest iteration of CellPhoneDB, what they do is that they filter. I believe they filter interactions based on the convolution results. So if they find that cell types that co-occur together in space based on the convolution results, I believe they actually run such location in the paper. They can then filter out real, let's say, interaction based on the spatial graph for the ligand receptor interaction analysis. And this is a two steps analysis, right? It's not one function at all, but it's really easy to integrate it here. I'm actually going to show something else in a second. So it's computing again the permutations on each ligand receptor interaction pairs against in each cluster, and then we have a plotting functions for doing that. So I'm gonna wait for it to finish. So you can of course increase the number of jobs in parallel and so on. We have, I think, other ways to increase the NUMBA performances by allowing for parallelization. But yeah, in this case, I'm just using 2 cores. I'm working on my Mac, on my Mac 2019. So all this computation is done there. For some of the things, I think you can scale really large. So for the neighborhood richness and spatial graph computation, you can scale for very large data, like hundreds of thousands easily. For the image part, probably you would have to have the compute clusters. So maybe a laptop is not enough. And I should also mention that in this case, we're using the default omnipath annotations for the analysis, but you can select specific databases, for instance. I believe we do show it in the tutorial. We do show it in the tutorial, I think. I know we have examples. Yeah. So here you can decide, you can essentially say, okay, actually I want only the annotations present in ServoDB. You can pass these interaction parameters and it only focus on one resources. For looking at more resources, you have to look into omnipath. And this is the database. There's a lot of things you have to look into here. Now I'm not super familiar where to look into it, but you should search for the ligand receptor interactions. So you see these are the databases, and you could essentially narrow down the analysis on some of these databases. Now we're doing it on all. And as oftentimes, unfortunately, this analysis returns a lot of hits, right? A lot of significant, potentially significant interactions. So now we have, despite the fact that I'm filtering by means and alpha value, right, the FDR corrected p-value, we still have a lot. Now, one analysis I want to show is how to do this, how to use the information for computing another type of interaction plot based on now the number of significant interactions that each cell type has, each spot has. And again, this I think is useful either if you have single cell level information or if you have the convolution level result, the convolution results for Visium. In this case, we don't have the convolution results, so I'm just going to show you how to do it, but you could integrate them essentially. So the point is, we have a neighborhood enrichment analysis before, right? We showed it before. So this is based on the spatial graph. Now we could compute the same type of interaction based on the number of significant ligand receptor interaction pairs that are found between each cluster. So how do we do that? It's actually quite straightforward. We save the results of the ligrec into adata.ans in squidpy. And this is a bit of complicated. It's like a dictionary of multi-index data frames, but let's just look maybe at the means. It's an index data frame, so you have the source and target and 2 clusters, right? So one way to do it would be, for instance, to filter out the interactions based on, for instance, p-value. So you want a p-value below 1.e⁻⁵, 1e⁻⁵, and that means above 2. This is just like a one filter, essentially. And then now we compute a mask array that masks essentially where these conditions are true. And then we sum over rows, and what we get is this is essentially the number of interactions. Now, in this case, you can see that it's just an array of essentially the 2 clusters concatenated. So we have to reshape it. We reshape it based on the cluster shape. So how many clusters do we have? And we have, I believe, something like 12 clusters, something like that. So these are the clusters like we're looking at. And then we save this array into the cluster interaction slot, and you can actually plot it. You can plot interaction matrices of any kind. So you can compute interaction matrices of different kinds, but this is a way to do it, for instance, and you can directly plot them with squidpy. And this is the results you will get. Now, each level essentially is the sum of the interactions. And you can see, for instance, that the pyramidal layers have a lot of interactions, counting interactions of linear receptor interaction now, significant. And this gives you a different view, sort of the interaction is going on compared to the spatial graph. Again, with the convolution results or with single cell data, this is probably more informative. And again, a lot of this actually will be shown by Anna later on much more advanced ways to do this kind of analysis. Yeah. So last thing I want to show is the spatially variable genes. So spatially variable genes essentially aims at finding genes that are spatially variable, not random on the tissue. And there are many ways to do it. In FLIPper, we have two ways, Morans or Gay-Ritchie. So these are spatial autocorrelation statistics, like very easy to compute, very fast. We have also more advanced one like Spatial from Alma, originally from Alma. And but there are other ways. I mean, Spatial DV, one of the most popular ones. Spice Mix, I think, and then some of the Spark. Spark is another great tool, but yeah. And the way I guess that I would maybe look at this type of analysis is another way to score genes that now, instead of being highly variables based on the expression, as is usually done in squidpy, is where they are spatially variable. So it's a kind of a feature selection step. And the results is the I is essentially the statistical computing by Morand with a bunch of p-values, both p-values for different type of tests. Tests based on normal distribution, normal assumptions, or based on permutations. And I think there are some problems in terms of power here, as in they're always, most of them are significant. I think it's because we're looking at many data points compared to like the standard uses that people use in statistics. But I would maybe look at essentially score them by the I score for Morans and and these are three genes to be found significant for this specific analysis for spatial variable gene selection. I guess I'm done. I think I have 10 more minutes. I didn't really show much on the spatial plotting, but I just want to show you that we can do more complex spatial plotting. So wait, let me move this. So we can plot multiple slides. So here, for instance, in this example, I'm creating two instances of the same slide just for visualization purposes, but you can have many more, like the one I showed, for instance, for nanostring. And the way you essentially save, actually, you know what, I'm just going to do this really quick. Oh, it's throwing errors. Okay, actually, I don't know what's going on now. Maybe. Ah, yeah, I think I know why. Because I have a data frames. This is an issue on ncem. Yeah. Okay, so I'm gonna try by regenerating the data set. Sorry for that. Maybe I'm just gonna copy the one here. Okay, and this is the type of plot I want to show you. So in this case, we are cropping and plotting multiple library IDs. Now, the first library IDs is this one essentially. The second one is this one. Now, the second one is subsample, just to show that you can have multiple ones. And you select how to plot it by using the library key. Library key is an entry here, which tells you isn't it here? Ah, sorry. In undata.ops, which tells you which that set D belongs to, essentially, which was the unique library D. And you can plot again the graph and so on and so forth. And as I showed before, this also works for the segmentation masks. So when you have, we have multiple slides essentially. And you can also select, for instance, whether to plot first the library or the colors. So now we're plotting genes and clusters. But you could also plot first the libraries. Sorry, this is the default. You could plot first the colors. So you have first the colors and then the two libraries. So on and so forth. Yeah, and this also works for spatial segment. Okay, I think I'm going to stop and answer the questions. Okay. Let me go through the questions. Is there a way to relate nuclear simulation engineering and spots so we know the exact number? Yes, there is, and we show it in the reproducibility repo of the paper and also here in the examples. Image. Yes, here. So in this tutorial here, extract segmentation features, we show that we are essentially extracting segmentation label here. So I'm not going to go through the tutorial, but basically we are extracting the number, the counts, and then the segmentation's intensity, segmentation summary statistical intensities on the segmentation masks only. So in this case, if you run this tutorial, essentially, here we are counting how many nuclei there are, segmented nuclei there are under each spot. So it goes from 45 to 0. And this is based on a standard watershed algorithm, which is maybe questionable in the way how well it works. But you could essentially plug your theory deep learning models and then use this for doing this kind of analysis. Can you compare classes comparing gene expression across different samples? Yes, you can. This is maybe standard scampy analysis. I would point you to the best practices or scampy tutorials for doing that. Do you have time to do a quick demo in interactive mode? Yeah, I don't think so. So the interactive mode is this one. Wait, no, tutorials. No, advanced. No, this one, Apari. Essentially, you need to have the data in the image and then pass the data to the image interactive of Apari, and that's what you get in Apari viewer. Now I'm doing a screenshot here for visualizing it on the docs, but you can work on this interactively. I should also mention that a lot of this is being rewritten now, and eventually we will have a separate plugin outside the repo that will rely on this new spatial data object for doing way more complex, for handling more complex data sets, basically, even multimodal, right? So maybe you have multimodal measurements on the same system across different sort of slides of different cuts of the biopsies, and you would want to look at them interactively, and this will be possible. It's going to take some time, so maybe say a few months, but we're doing a lot of rewrite on this side. Manually assign class and machines, you can, yeah. You would have to use the shape things that we showed in the slides for Napari, but I think it's very tedious, so I don't think we have a fast way to do it. By version element case where gene expression based classes not exactly overlapping with image based. Yeah, so this is what I showed here, I guess, but this is not super interesting, maybe settings, but this is one way to look at it. Was it here? Yeah. So now the clusters on the image are very different than clusters on gene expressions, and essentially it's up to the analyst to understand why that's the case. For the H&E, I guess I would argue it's mostly due to cell densities, so how many nuclei you have, and some maybe like the high level morphology features. When fluorescent images, you could only go down to like the protein expression, basically, because you have the fluorescent marker, and then you can ask more interesting questions. If you have segmentation masks, even better. Nanostring is a great technology to do that, and we're gonna have a tutorial coming up soon on nanostring. How important is segmentation analysis for spatial data? I mean, that's a good question. I would argue it's quite important. I think it's interesting to look at. I don't know. We really kind of stress that the image that you acquire in spatial economics should be used as a modality. So not only for visualization, but you should actually analyze it essentially. And segmentation one is one of the analyses that can be done. I think that some tools for the convolution, for instance, I believe Search Location does, maybe Tangram does also, and maybe others. They use the information of how many nuclei you expect. I think it's either an average. It's basically like a prior that you have to set, and you could use it by directly segmenting the tissue and computing it. and then pass it to the deconvolution method. So I think, yes, it is useful and it has potential impacts, for instance, in the convolutions. I'm not familiar with other analysis that actually leverages this directly, but yeah. Plan to include GPU computations. That's a good question. GPU computation in terms of analysis. So you can already do like plug in, for instance, image containers for TensorFlow, PyTorch. The analysis on GPU, that's a good question. I don't think so. No, I don't think so. We have used Dask and Dask image for doing basically analysis on chunks, so out of core. So we don't load the whole image together, but we load chunks and we operate on chunks. For GPU, I don't. Maybe they do. Maybe they do have actually ways to do it directly by using a library. But I need to look into it. Is there a way to integrate imaging mass spectrometry data? Yeah. Well, you could. This kind of analysis can be done also in imaging mass spectrometry. I think actually Alma as well has very nice results in imaging mass spectrometry, and she will show you how to use these tools in that case. But for pre-processing, I would argue not. Maybe for pre-processing, you should use specific tools that go essentially from the raw image, because there is a lot of things that needs to be done, right? Oftentimes it's illumination correction, stitching, any type of like pre-processing, smoothing, segmentation, and so on and so forth. And for these, I think there are better tools than the image container. If you have multiple samples in the linear receptor analysis, which normalization and FTR correction would you recommend to use? Multiple samples in the linear receptor analysis. I think you don't need to. I think you can just look at them together. The same way as that, I think in CellPhoneDB, they also do it by just aggregating them. I think, I'm not sure, but I don't think, I mean, for the specific test that they use and we use, I don't think that you gain any information by adding essentially the batch, because it's just permutations, it's just like shuffling essentially. Is there a tutorial for single molecule data, single cell or mini bulk data? Yeah, there is. We have tutorials on this kind of analysis on SecFish, for instance. And so selection is like SecFish here. We're coming up with nanostring and BaseGen very soon. I think that will be maybe interesting. Yeah. Is there any plan to integrate assessment methods such as cell pose or vector? Yes, there is. Actually, there are already, as I mentioned before, we have cell pose is already there. You can use cell pose right away. You can plug it right away. Oh man, I forgot that. Yeah. Yeah, base or not, although I think there is, so we want to start looking at transcripts level informations and doing this kind of analysis on the transcripts. So Baser looks at the transcripts coordinates, right? And then builds these pseudo cells, densities of transcripts and counts the transcripts there. There is other tools. I think SAM is one of them. I think there is something else from Danaper recently. And we definitely want to include those. We need a better data representation first to start with, but we will eventually. At least maybe not directly in squidpy, right, but maybe like again part of the ecosystem. It's relatively straightforward for the analyst to plug this step before a squidpy analysis or before an ncem analysis. Yeah. We just play again. What is the scale argument when image features are calculated? Yes, the scale argument is the context exactly. So it's basically like how. So we always have. So each spot. I'm going to show it. I think if I show it on an image, it's better. So each spot, right, it's not a scatter plot, it's actually an area, right? A specific area defined by a diameter and a centroid on the tissue. So if you use scale equals to 1, you're essentially just cropping the array on this under this specific area and computing the features there. If you use scale equals to 2, you're essentially looking at a larger area. Or if you use scale equals 0.5, you're looking at a smaller area with respect to the centroid. So yeah, it is different. CellChatDB for NSI would be cool. I don't think we have an easy way to access the database to have like probably. I mean, I'm sure the developers have developed it, but we don't have it in squidpy, so I don't know whether it's going to happen soon. Although I wonder if Omnipad has it. If Omnipad has it, we should have it. No, they don't have it. Yeah. So maybe it's something we could talk about with Omnipad. I have seen a tutorial where mitochondria and hemoglobin genes were filtered out at early stages. Yeah, this is again something that I don't have strong sort of suggestions or opinions. I think mitochondrial genes are often filtered out. So cells without mitochondrial counts are often filtered out because they are deemed to be sort of dying cells or with a metabolism that is a bit skewed from the rest of the population, or they are under stress or something like that. although I think that in some cases people don't. I think, like, for instance, for some tissues like the liver, I believe, since the liver is very highly metabolically active, then by filtering by mitochondrial counts, then would be a problem. But yeah, I don't know. But again, this is something that it's not, let's say, I don't know much about this, so I don't want to like give suggestions. Is it possible to compare changes, especially in rich genes, with samples conditions? For example, check there is a shift in the spatial domains. Yeah. So this is also a very big thing that we were working on actually with Alma and David, and there is lack of analysis that look at spatial variability, or basically spatial variability across conditions. I think some of it will be shown by Alma. Alma has a very nice ways to look at this kind of thing with spatial differential expression analysis after registration. We are looking into how some of these statistics can be extended to account for the conditions for doing this differential analysis. But we have nothing yet. But it's something that we're looking into, because indeed, oftentimes analysts don't have work with one slide, right? They work with multiple slides, and maybe multiple slides across conditions. So it should be accounted for, yes. I think I'm right on time, 3 o'clock. Are there more questions maybe on the chat or on Zulip?
No, I don't think we have any more questions, but feel free to ask maybe 2 more if anyone has a question.
Yeah, anybody has more questions? Yeah, I can also open Zulip, but I don't think there's anything.
And I additionally would like to highlight that we will stay active on the Zulip throughout the break. So if you have questions or issues installing the tools, just raise them in the break, or at any time we will keep this Zulip open even after the workshop.
Yeah.
And also the slide.
I see some questions on Zulip. So one is on the interactive mode work well with Jupyter Lab. If you're for SSH forwarding to a local server, no, that's a problem with Napari. I think there are no solutions in Napari, but I didn't explore them. I think maybe refer to Napari issue tracking or Napari Zulip. Actually, no, we are in Zulip, but there is also, okay, maybe I can share the screen. So another great point. So we have Zulip, right, for SCVers, but this is also Zulip for Napari. And that's a great place to ask questions for Napari, for instance, also Zulip. So yeah, unfortunately, I don't have an answer for that. I should also maybe mention that there are other ways to do this interactive visualization. One of them is called tissue maps, and it's really, really great. Tissue maps, I think. Yeah. And this is, I think this is easier to work with for if you are in a server, and they have pretty good documentation or tutorials, so definitely recommend that. Another one is called BTSC, and this is something I'm a bit less familiar with, but I think it's also really good. And they also have Python. They have Python. They have a way to run it in Python basically. And I believe also they load data directly. So you see Jupyter Lab. This is an example of Jupyter Lab. This is an example of RStudio. So maybe these are additional tools. Yeah. So interactive visualization, there is a lot of stuff out that I think it's relatively straightforward to plug into what you already have in terms of data format with data. I see Alma has points on normalizations. Yeah. And there's a tutorial and a paper. And I don't see anything else on Zulip. And on Slido also, that's it. And chat. That's it also. Yeah. So how should we do it? Should I stay online for another maybe 5 minutes and then log off and just answer on Zulip before your next talk, Anna? Your talk starts at 3:30, right? And no,
yeah,
David.
So I would keep the 30-minute break so people can relax and have a coffee or enjoy their break. So I would resume with the schedule at 3:35. So exactly in half an hour. We will stay for a little while in the Zoom call to ask questions to Giovanni, or again, as I said, on Zulip, Slido, or any channel you would prefer. Then yes, then see all of you again at 3:35. Looking forward to the next session, which will be hosted first on the introduction by David and then by me on our tool ncem.